In the Oxford Museum, Apidae are found in the Hymenoptera collection, which contains bees, wasps, and ants. The collection has 3,281 specimens in total, with 44 from the Apidae family. The specimens were collected from across the world, including Neotropical, Palearctic, and Oriental regions, and date back to the 19th century. The specimens are primarily staged or direct pinned for display. The pins used were formerly copper, however due to the damage caused to the specimens, iron pins are now used. On our viewing of the virtual tour, we took an interest in the bumblebee specimens housed within the Hymenoptera collection. Further research into the subfamily and family led us to Apidae, which our presentation has focused on. Bees, and therefore Apidae, are all related to carnivorous apoid wasps. These predatory wasps gave rise to Apoidea, which is a superfamily consisting of modern-day ants and wasps. Melitosphex burmensis from the early Cretaceous is known to be an extinct lineage of pollen-collecting Apoidea sister to modern bees. One cause for diversity is hypothesized to be environmental adaptations. The switch to herbivorous diet allowed a new ecological niche to be created. Lineages transitioned from a carnivorous diet to a more specialized herbivorous diet of pollinivory, giving way to the diversification of bees. Research has suggested that behavior of giving young pollen or nectar is a trait that was passed on from ancient carnivorous wasp ancestors, proving the transition from a carnivorous diet to one of pollinivory. Behavioral adaptations such as eusociality and morphological adaptations due to coevolution that allowed bees to become specialized to particular flower types are also reasons for diversity. Case studies of this are the genera Rediviva and Diacia. Rediviva bees evolved in response to oil-producing floral spurs in the snapdragon flower. The bees have velvet hairs on the end of their extended front legs which soak up the oil and mix with pollen. Bees have also evolved specific lengths of tongues. Due to this morphological adaptation, the 7 bee families have differentiated into 2 main groups, long or short-tongued. The fossil records suggest that short-tongued families are Coletidae, Adrenidae, and Halictidae, and may have arisen later than the Melitidae and the long-tongued families, making the long-tongued clade the sister group to all other bees. Overall, a non-conclusive result for the evolution of bees, due to most whole genomes not being available for all bee species.
Apidae is the largest, most specious, and behavioral diverse family of the bees, apid. It comprises social forms, solitary, socially parasitic, and a high percentage of kleptoparasitic species. This family of long-tongued bees includes around 6,000 species, comprising 35% of the world's bees. There are conflicting studies on whether this family is divided into 3 or 6 subfamilies. However, for the purpose of this presentation, we have presented the diversity of Apidae within the 3 subfamilies Nomedinae, Apinae, and Xylocopinae. Studies have shown that the 3 subfamilies have diverged from one common ancestor, with Xylocopinae as the most basal lineage. This theory is supported by the shared characteristics of Apidae that is present in the ancestral species, such as social behaviour and the long adult lifespan. However, the evidence supporting Xylocopinae in this theory is lacking in comparison to the other 2. This means that future studies using molecular analysis in these subfamilies, especially Xylocopinae, could see a change to this theory, and tribes may move between each of the subfamilies.
The social behaviour of bees evolved 90 million years ago, during the Cretaceous period. The family of Apidae consists of a wide array of social behaviours, from solitary orchid bees to primitively eusocial bumblebees and highly eusocial honeybees. Social behaviour in bees has been suggested to increase the rate of evolution and also provide a greater variation between eusocial bees. Eusociality is the climax of social organisation, with characteristics such as cooperative care of broods and division of labour. This behaviour can be found in bumblebees, stingless bees, and honeybees. The vast majority of the total bee population consists of solitary bees at roughly 90%. These bees aren't part of a colony and forage as a single individual. Orchid bees show solitary behaviours, but are also known to show communal social behaviour.
The species in this family show further diversity, with some species being parasitic and others not. There are 2 major types of parasitism in bees, being kleptoparasitism and social parasitism. Kleptoparasitism is more often seen in bees, especially within this family. Kleptoparasitism involves the parasitic bees invading the absent nest of pollinating bees to insert their eggs into the brood cells of the nest. The parasitic larvae will then be cared for by the host bee. Kleptoparasitic bees can be found in both subfamilies Apidae and Nominidae, and are absent from Xylocopinae. The parasitic nature of these bees is recognised as a behavioural-derived character, but morphological adaptations have also developed to aid these bees in acting as parasites, such as an increase in armoured skin. In most kleptoparasitic bees, their larvae also shows morphological adaptations, in the form of large mandibles which allows them to kill host larvae and any other kleptoparasitic larvae that is in their way to enable them to receive as much pollen as possible from the host so they are allowed to fully develop. Cardinal et al. Showed using molecular analysis and fossil records that kleptoparasitism evolved from a single ancestor for most bee species in this family. However, for all kleptoparasitic species in this family, there are 4 different origins of kleptoparasitism, which is most clearly shown by orchid bees in the Apidae subfamily. They were able to estimate the origin of kleptoparasitism to be around 95 million years ago. It should be noted that with the emergence of new data, this estimation is variable to change. The Nominidae is the second largest subfamily and has 10 tribes with around 1,300 species found across the world. This subfamily is entirely kleptoparasitic, which is also referred to as the cuckoo bees, and they have a high diversity both within the subfamily and compared to the other 2 subfamilies. This is shown by both morphological and behavioural derived characters. An example of a morphological difference is the apical metasomal characters, which are used to inject the parasite's eggs into the host nest brood cells. All bees in this subfamily lack the morphology needed for pollinating. This has resulted in scientists assuming they are wasps upon first inspection, as you can see in the image on the right. The phylogenetic relationships of this subfamily are extremely complicated and are ever-changing as more research is done, as is evident by a study done by Rosen in 1996.
The subfamily Apidae includes the honey, bumble, stingless, orchid, and digger bees. It is estimated that there are around 3,500 species of Apidae worldwide, which can be separated into 19 recognised tribes. Apidae includes the highly eusocial tribes, Corpulate Apidae, which can be seen in the Oxford Museum collection. This contains the tribes Apini, the honeybees, Bombini, the bumblebees, Euglossini, the stingless bees, and Meliponini, the orchid bees, with fossil evidence showing these tribes are all related to each other. The pollen-carrying structures found on the tibia on the hind legs are unique to the females of the Corpulate Apidae, with these bees playing a vital role in the pollination of crops around the world, as well as being used as a model to understand the diversity of social organisation in insects. Apidae also contains several kleptoparasitic tribes, such as Ossorini, a bee solely found in the Neotropics, where it lays its eggs in the nest of other bees. Certain behaviours that are rarely seen in the other subfamilies of Apidae can be found in the bees of Apidae, such as the solitary tribe of Eucerini, in which the males can be found in large sleeping aggregations on leaves, or the neotropical tribe of Centrodini, that collect floral oils to feed their larvae and line their nests. The exact characteristics that cause so much diversity within the subfamily of Apidae is still unknown. However, fossil records, biogeographical studies, and morphological evidence indicates that the sister groups Apini and Melipini form the basal branch of Apidae.
The Xylocopinae subfamily, commonly known as carpenter bees, make their nest in wood, stems, or pith. They are the smallest subfamily and most basal lineage within the Apidae family. It contains 5 tribes: Xylocopinae, Ceratininae, Manuelinae, Allodapinae, and the extinct Borealodapinae, known from specimens found in amber. Each tribe contains only a single genus. There are approximately 300 specimens in total. The precise relationship between tribes, despite investigation using molecular data, remains controversial. Mostly, Xylocopinae species are solitary. However, some species in the Allodapinae tribe are eusocial.
The coevolution of bees and angiosperms is a perfect example of adaptation and specialization. Both bees and angiosperms affect each other's evolution through selective pressure, as they are interdependent upon one another. The concurrent rise of bees alongside the diversification of angiosperms may have been the driving factor in the rapid speciation of angiosperms. A study done by Miller-Strootman shows that floral resources are on the decline due to warmer summer periods, which has benefited the generalist foragers rather than the specialists. Results from this study show a decrease in the tongue length of 2 species of bumblebees, over a 40-year period. There is a larger degree of specialization in the long-tongue bees compared to the short-tongue bees, making them more susceptible to environmental change. This study shows that lower floral density has been the driving factor for the evolution of the bee's tongue length. Climate change can potentially adjust the foraging behavior of bumblebees at an individual level.
To conclude, this video has shown that the phylogenetic relationships of the Apidae family is ever-changing as new experiments are performed. The diversity within this family is a result of different behavioural, environmental and morphological adaptations. We also reviewed a study which shows the complex coevolution between angiosperms and this family. Thank you for watching.